Hello, my name is Himai Durrani, and my project is titled The Role of HOXA11 During the Decidualization of Endometriostromal Fibroblasts in the Human Uterus. I worked with Eric Rickenbach at Yale University's Wagner Laboratory. I was never really that interested in the subject of biology, but after applying for an internship at Yale University's Wagner Laboratory, I decided that molecular biology and genetics might be some of my main interests in research.
Through our school science research program, I applied for the internship for the Wagner Laboratory. Despite my interest in physics and astronomy, I decided to apply, and I found myself interested in new subjects like genetic engineering and evolutionary biology, for which I am ever grateful. At the lab, my mentor introduced me to my research topic and a slew of literature review that I used in eventually coming up with a research question and subsequent research project. I was subjected to official Yale University safety training, and then my mentor taught me how to use different equipment in the lab, like centrifuges, pipettes, and PCR machines. I was highly independent in the conducting of this study other than the basic supervision, as the internship was designed to allow me to discover my potential in a laboratory setting.
At the lab, I was heavily independent in coming up with research project and question. I decided after going through a couple of papers that Hox genes affect embryonic implantation, progesterone encourages positive uterine health, and endometrium-related infertility is correlated to decreased HOXA11 expression, which meant that my questions and answers lied with understanding more about HOXA11's importance and using the CRISPR-Cas9 system to effectively investigate real effects of an edited version of this gene.
Additionally, the project was in line with the goals of Professor Wagner, who had been requested by the Cincinnati Children's Hospital to further investigate the CRISPR-Cas9 genome engineering system. HOXA11 is a protein coded by the HOXA11 gene. It has a regulatory role. Decidualization is any change to the uterine lining due to progesterone, including increased blood cell proliferation and the changes required for a uterus to accept pregnancy. The HOX genes and proteins are related to female reproductive health, but the specific role of HOXA11 is unknown in decidualization. Due to the goals of the lab, we decided to employ the use of the CRISPR-Cas9 genome editing system that would be able to invalidate the HOXA11 gene and then subsequently tell us about changes in decidualization due to that change. CRISPR-Cas9 is a prokaryotic immune response found in Streptococcus pyogenes. The method lacks efficiency and requires proper design, but by perfecting its use now, it can be better utilized in later projects and applications. A series of assays were utilized to properly investigate this question, including a culturing of cells and conformation assay, a T7-Endo1 assay, genetic mutation of cells, and analysis of said cells.
This project is nearly complete as I worked at the laboratory over the summer for 6 weeks with a workday schedule, and I've continued working there over the school year with lesser hours. With the knowledge of efficiency of CRISPR systems as well as the tangible effects of an altered HOXA11 expression, much can be applied in the future regarding how to target the gene in order to properly remedy certain ailments, including genetically linked cancers. We've yet to perfect the CRISPR systems, but its practice is sorely needed in an era of increasing disease and potential genetic remedies.